Okay, we're live. So welcome to another event for Bilacura. This is the Understanding Cancer session. So it's my pleasure to introduce Dr. Florencia Pascual, who is a research scientist at the National Institutes of Health. Florencia received her PhD from Rutgers University and then did a postdoc fellowship at University of North Carolina at Chapel Hill. Before transitioning to her current position at the NIH, where she studies how metabolism affects development. As many of you know, Florencia is passionate not only about dance, but also about science and science communication. And she's an active member of our local dance community. Her long-term goal is to combine her passions to help further understanding how dance and yoga and meditation affect our bodies and our brains at a molecular level. So today, Florencia's presentation is going to be on understanding cancer, and it will complement the presentation you heard earlier from Emily Mathura. It will focus primarily on some basic information for non-cancer scientists and non-cancer biologists, understanding the types of cancer, how to prevent them, how to screen for them, what kinds of treatments are available, and the role that basic research plays in some of these things. So without further ado, I'll turn things over to Florencia.
Okay, thank you, Daryl, and hello, everyone. And yes, today I'm going to be telling you about cancer biology. So I will actually be going over the basic of cancer. See what I did there? And I wanted to give you my perspective as a research scientist, because a lot of you may know me from the dance floor, but you might not know that I am actually a research scientist by training. And so I wanted to give you my perspective of why it is important that we continue to fund cancer research so that we can understand how cancer works and therefore treat it or prevent it. So I can't see the slides, but if we can go on to the first one. There we go. So, philosophy aside, we are basically a collection of cells. And cells constitute the fundamental unit of life. If we were amoebas or a single cell organism, life would be pretty straightforward. If we lived in a nutrient-rich place, we could absorb nutrients and oxygen from the surrounding environment, and we could get rid of waste by diffusion. However, next. Most of the humans we know, not all of them, but most of them, are not amoebas. So things aren't quite so simple for a 30 trillion celled organism like human beings. And in complex multicellular organisms such as humans, we have specialized cells that organize into tissues. And these are groups of similar cells that work together on a specific task. And organs are then structures made up of two or more tissues that are organized to carry out a particular function. And groups of organs then organize together with related functions to make up the different organ systems. And just as the organ systems work together to accomplish specific functions, the different organ systems also cooperate to keep the body running, or in our case, dancing. And so I think we have established that we are in fact a collection of cells. And the few million cells that we have today will be different from the few million cells we will have next week. So it is this constant cell turnover that keeps our bodies functioning. Next. But this only works, however, when it occurs in a programmed and regulated process at the individual cell level. And that's where I will be spending most of my time today. Next. So what makes a cell a good cell? Let's go over the processes that make normal cells behave the way they do. Next. Although animal cells vary considerably depending on what function they have, they all exhibit specific structural features and contain a number of organelles that are suspended in the cytoplasm. Cells are surrounded by a plasma membrane, which acts as a protective barrier from the exterior, but also regulates what comes in and out of the cell and serves as a means of communication with the space outside of the cell or the extracellular environment. Animal cells also have a true nucleus that's enclosed by a double membrane. And inside this nucleus, we have the majority of the cell's genetic material. And we store it in the nucleus, but we also process it in the nucleus. It is this processing that coordinates most of the cell's basic functions. The ER, the endoplasmic reticulum, is a network of membranes that is connected to the nucleus. And this is where proteins and some lipids are synthesized. And proteins that are synthesized in the ER then travel to the Golgi apparatus, which is sort of the mailroom or the distribution center of the cell. And here the proteins and some lipids get modified, they get sorted, and they get packaged for transportation to either other locations of the cell or outside of the cell. All of these processes are powered by mitochondria, the powerhouse of the cell. I'm sure you've heard that expression. And in mitochondria, cells break down nutrients to produce energy in a process called cellular respiration. And finally, there are dynamic networks of protein filaments and microtubules that give shape to cells, but also allow for transportation within the cell and for cell movement. Next. Another feature of good cells is that they exhibit a rapid exchange of chemical materials with both the external environment as well as within the cell itself. So metabolic substrates such as carbs, lipids, and protein, basically the food of the cell, are converted into cellular energy, signaling molecules, but also structural components of the cell. And on the bottom part of the screen, I have a, believe it or not, pretty simplified scheme of the major metabolic pathways that occur within a cell. And I wanted to show it to you so that I can illustrate how all of these pathways are interconnected. So one metabolite from one pathway might be used for another one, but also I wanted to use this opportunity to tell you that metabolites or byproducts of these metabolic pathways can also be used for different functions, such as other cellular processes that have nothing to do with energy production within the cell. Next. All cells have a life cycle or lifespan, and this includes periods of growth, rest, and respiration. And so this diagram here depicts very briefly what the cell cycle looks like. And there's 3 initial stages where the cell grows, it copies its DNA, and it gets ready to divide. And then in mitosis, the cell stops growing, and the newly copied chromosomes are divided into 2 to make certain that each new daughter cell gets a full set of chromosomes. And when the mitosis is complete, the cell divides, and the new cells are now ready to go through the cell cycle again. And in this diagram, you can also see an example of one checkpoint that is used by cells to correct any potential replication mistakes that may have happened. So we talked about how our cells are in constant turnover. So we're making billions of cells every day, and that creates a lot of room for mistakes. So good cells or normal cells respond to these checkpoints that exist at different stages of the cell cycle. And what happens when they get to those checkpoints is they wait for signals from the exterior that tells them, keep going, continue growing. And these are called growth inducers or factors, or signals that tell them to stop, which are growth inhibitors. Next. But good cells don't only know how to or when to grow and divide, they also know when to give up. And they know when they have given their all, and it's time to make room for a new set of healthy cells. And they died in a controlled, regulated fashion in a process called apoptosis. This process is important for maintaining constant cell numbers. So if we're constantly making new cells, we need to make sure that we are eliminating cells that we don't need. It also provides a means for eliminating damaged cells. And it plays a very important role in development. So for example, it eliminates the tissue between the digits during embryonic development. And so during this process of apoptosis, the normal cell undergoes various changes, and some of the major ones are that its genetic material gets cleaved and condensed. There's a blabbing of the plasma membrane, the cell shrinks, it fragments, and eventually it is cleared by specialized cells. Next. And another feature of normal cells is that when they develop harmful mutations or they become damaged by either microbial infection or mechanical stress, they expose a find me signal on their surface. And I drew it as a little pink thing on the cell surface. This is a cell's find me signal, and this signal is recognized by immune cells who actually engulf the cell or eat it and degrade it into its components so that they can be reused for to support the growth of other cells. Next. And one of the final features I believe I will talk about for good cells is that they don't travel. Even though tissue formation during embryonic development or wound healing or for the immune response sometimes requires orchestrated movements of cells from one direction to another, most of good cells in our bodies do not spread beyond the area where they belong. So for instance, your lung cells will not travel to the heart, even though they're in very close proximity. And this is because cells secrete cell adhesion molecules that make them stick to their neighboring cells, but also to the surrounding extracellular matrix. And this is what keeps them in place. Next. So we've seen how organisms maintain their homeostasis or their healthy equilibrium through coordinated cellular processes. But cancer is the most devastating consequence of a loss of control of any of these processes that we talked about. And so when the body loses the ability to control whether a cell grows or it replicates or it's going to become specialized, this cell acquires the potential to take over the rest of our body. So simply put, cancer is good cells gone bad. Next. So now that we know what constitutes a good cell, let's see how good cells and bad cells compare, but also let's look into how knowing this about both good and bad cells can help shape the therapies that we develop to treat cancer. Next. So let's start talking about structure like we did for normal cells. Cancer cells often don't exhibit the same structure as normal cells. They have a lot more variability in cell size, so they can be either larger or smaller than normal cells. And they often have an abnormal shape, both of the cell itself, but also the nuclei, which is shown in this little diagram here. They also have a reduced stiffness, so they're more easily deformed than normal cells. And they generate increased contractile forces and are also very strongly influenced by their environment in terms of their biomechanics. So we can use these characteristics to distinguish cancer cells from normal cells and to separate highly invasive cells from cells that are not as invasive. And we can exploit this to develop either diagnostic or prognostic applications such as nanorobotics, or some of you might have heard high-intensity focused ultrasound that can target cancer cells specifically and bypass normal cells. Next. Another feature of cancer cells is that they don't have a normal metabolism, so they don't obey the law and order that normal cells follow when it comes to metabolism. And I showed you a pretty complicated diagram before, but here I'm zeroing in on the most popular metabolic pathway that cells use. And this is how cells obtain their energy in the form of a molecule called ATP. And they do it by breaking down glucose via a process that's called oxidative phosphorylation. And some of their energy also comes from an anaerobic process that occurs in the absence of oxygen that's called glycolysis. But most of the cell's energy comes from oxidative phosphorylation. However, cancer cells actually obtain their energy through glycolysis, even though there is even when there is oxygen present. And this is a phenomenon called the Warburg effect after the person who discovered it. And for a long time we thought that maybe cancer cells had defective mitochondria, which is where oxidative phosphorylation occurs, and that was why they couldn't obtain energy through this process. However, we now know that that is not the case. And so our thinking now is that actually the reason why they obtain energy through glycolysis is not necessarily because of the energy itself, but actually other metabolites that are being produced throughout this pathway that can then be used to build macromolecules that promote cell growth, which, as we know, is very important for cancer. So even though we now know that cancer cells can adapt to changes in their metabolic substrate, they're not as inflexible as we used to think they are when it comes to what substrate they can use. We can actually harness this reliance on this specific pathway to target cancer cells specifically versus normal cells. Next. Another feature of cancer cells is that they don't stop growing. And as we saw earlier, normal cells rely on growth factors and growth inhibitors that tell them to either continue growing or to stop growing. Cancer cells continue to divide and grow in the absence of growth factors or in the presence of growth inhibitors. And this lack of checkpoints means that these cells have a lot of genomic instability. So if cancer cells don't stop every now and then to check for possible errors in their replication of the genetic material, then this creates room for a lot of mistakes in their DNA or mutations. And so some of these mutations are driver mutations, which means that they drive the transformation of the cell into a cancerous cell. And others mutations are what people call passenger mutations. And this means that they don't affect the function of the cell at all. And these passenger mutations are very specific to the tumor itself and to the person that has the tumor. So we can use those passenger mutations in personalized medicine as potential targets for cancer therapy. Next. Another interesting feature of cancer cells is that they are immortal. So we know that normal cells are immortal, they have a lifespan, and this is in part due to a shortening of a protective structure that's found at the end of chromosomes. That went by really fast, I apologize. That is called the telomeres. And every time the cell goes through cell division, this structure gets shortened, and eventually when it's too short, the cell can't divide anymore and it dies through apoptosis, as we've seen before. The cancer cells, however, have found a way to circumvent this process by activating an enzyme called telomerase that elongates these structures at the end of chromosomes, so they essentially become immortal that way. Another way, which is shown on your screen right now, is that cancer cells can evade apoptosis. And so actually this particular feature of cancer cells is what most of cancer research has focused on for the past 30 years, is in trying to develop therapies that promote the activation of apoptosis in these cells. Where novel therapies are going now is in targeting apoptosis in combination with other cellular processes, such as the immune response. I think the slide might be stuck. Can you hit next, please? There it is. And next. And unlike normal cells, cancer cells can actually evade or trick the immune system. And they do this by either escaping the detection through the lack of those signals that normal cells have on the cell surface. So they have fewer of those signals, or the signals are masked so that immune cells can't recognize them. But also they secrete chemicals that inactivate immune cells that come to the scene. And some of the novel immunotherapies, so therapies that target specifically the immune response, are focused on enhancing the function of some of the cells in the immune system, so allowing them to better recognize cancer cells, but also it inhibits the production of these chemicals by cancer cells that blocks the immune response. Next. And the last two features that I will talk about are what makes cancer as dangerous as it is. And that is, one, it can create its own blood supply. And so normal cells cannot spontaneously promote the formation of a blood supply, but cancer cells can. And they do this in order to shuttle or transport nutrients and oxygen to cells that are within a solid tumor that otherwise would not have access to those nutrients. And so that is what promotes tumor growth. So what we can do to fight cancer in this way is to use angiogenesis inhibitors, and that is block the formation of or the creation of a blood supply. Next. And finally, and this is potentially the most dangerous feature of cancer cells, is that they actually can travel and they are able to establish colonies in other tissues. So you can have benign tumors, as shown on the top half of the slide, and these are cells that are contained within the tissue where they originated, versus an invasive cancer cell, which can rupture through the tissue, enter the circulation, and travel to other tissues where it can start growing again. And most of the time, these new tumors are actually very different from the primary tumor, which makes metastatic cancer very hard to treat. So because some of the cells in our body require the ability to move in order to function properly, such as cells in the immune system, we can't really block cell migration in order to treat cancer, but we can enhance the migration of some cells, such as the immune cells, so that they can better migrate into tumors to treat cancer. Or we can also enhance the formation of those junctions between cells and between a cell to the matrix, so that the cancer cells can stay fixed in their place of origin. Next. So what causes these cells to undergo this transformation from a good cell into a bad cell? And we know now that cancer is a result of the accumulation of mutations in genes that control all of the basic cellular processes that we talked about. So these mutations can occur either inheritance.
Okay.
Everything okay? I can hear you guys. They can occur either, they can be inherited, or they can be caused by exposure to carcinogens such as tobacco, radiation, some viruses, and pesticides. But we also know now that lifestyle can also be a potential risk factor in the development of cancer. So, for example, high-fat diet or lack of exercise can contribute to obesity, and obesity is associated with risk with cancer. Next. And so how do we study cancer from a basic research perspective? We used two main study models. We do in vitro and in vivo studies. And so in vitro, we use immortalized cell lines. We take advantage of the ability of cancer cells to continue propagating and growing in order to grow them on a dish in our labs. And we actually use cancer cell lines for studies in other fields of research that are not related to cancer, precisely because they produce basically a limitless amount of biological material that we can take advantage of. But we also need to rely on animal models, because even though we can derive a lot of information from cell studies, we can't understand how a tumor might interact with other cells surrounding it in a 3D way, or how it could relate to the environment that surrounds it. So in order to do that, unfortunately, we need to go up a level of complexity and look at animal studies. And so we can use animals that have mutations that were either genetically engineered, or they were derived from exposure to specific carcinogens. And some of the more recent studies actually use xenograft models, which is we can take cells in culture or cells that were derived from a cancer patient and inject these into an animal and see how they behave. So that effectively we're combining in vitro and in vivo models. And some of the fancier and more modern, I guess, ways of studying cancer is to use 3D bioprinting. So we can print tumors or tissues and understand how they behave. And as also mathematical modeling or computational modeling to understand cancer. Next. And so I hope that by now I have convinced you that the reason why we can treat cancer today is because we understand it much better than we did a few years ago. And that is because we've done a lot of research which was funded by federal grants, but also by nonprofit organizations like La Cura. So we need to keep the funding going. And even though more people are beating cancer today than ever before, and survival has doubled in the last 40 years, the risk of cancer is still very high, and it's actually increasing. And we all have been directly or indirectly affected by cancer. So we know all too well that this risk has continued to increase. And this is due to multiple reasons. It could be because our cancer-specific screening is more available and a lot more sensitive than it ever was. So now we're able to detect it more often and faster or sooner. Older people also get more cancer or cancer most often, and we're getting older. We are more likely to be overweight or obese than in decades past. And I've mentioned a few times that obesity has also actually been linked to increased cancer risk, but also to decreased effectiveness of cancer therapies. And there are also other certain cancer types that are on the rise. So we must continue to fund cancer research, and that is why non-federal sources of funding, such as nonprofits like Baila Cura, are so important. Next, which brings me to my last slide, where I want to give a shout out to the event organizers for asking me to tell you a little bit about cancer biology, but mostly for coming up with new and fun ways to continue funding cancer research. And thank you all, for the ones of you that are still on the other side of the screen. I hope to see you on the dance floor soon, and I will take.
Great. Wonderful presentation. You really have a nice way of making complex stuff sound understandable and simple.
Thank you.
While we're waiting for some of the questions to pop up on the bottom, maybe you can.
I see.
I don't see there. Okay, actually, Susie posted the first question. So, can differences in cancer cell metabolism be exploited? I guess in terms of exploiting for therapy.
Yeah, and so I talked a little bit about that on the metabolism slide, but we know a lot more about cancer cell metabolism than we used to, and we continue to learn more about it. So while we don't have a specific way that we are targeting it today, we can use it in conjunction with other therapies to make them more efficient. And we hope that the more we learn, obviously, the more we will discover other things that we can use specifically for cancer cells in terms of metabolism.
Yeah, and you want to say a little bit about personalized medicine while you're there. I mean, nowadays oncology therapy is all based on understanding specific types of cancer, and how does that work? Explain how that works.
So I covered a little bit about that in one of my slides, but basically, not all cancers are created equal, and that is very different from what we used to think. So we used to treat cancer with there was one treatment fits all kind of mentality, or that was what we had available. Now we know that because these cells are so susceptible to mutation, and these mutations are very random, each cancer or each tumor is very much its own planet. So we can take advantage of the technologies that we have now, or we can actually basically sequence every gene in a specific tumor and see what gene is defective, and we can target that gene specifically, which is what essentially personalized medicine is about. And we know that some of those genes are specific only to the tumor, so we wouldn't be affecting the patient's cells, for example.
Yeah, so I think Lulu says hi, I guess.
Yeah, I can see that. Hi, Lulu.
So let's see, Matt asks, so what is the
Warburg phenomenon?
The Warburg phenomenon is. Help.
And that is it's what happens when cancer cells use the metabolic pathway that normal cells don't usually use. And that is that we essentially think of it as not an efficient way of producing energy. Okay. But we now know that is not the only reason why they probably use this metabolic pathway, okay, or prefer this metabolic pathway.
So the other Susie says, will immunotherapy be a turning point in the fight against cancer?
So I think in recent years, it's been one of the most talked about because it's one that shows a lot of promise, and we still don't understand a lot or haven't been able to harness it as effectively as we thought we would. But definitely using it in conjunction with other therapies that target, for example, apoptosis, which we've been using for a long time. I think that's where they're starting to see that it's a lot more effective for some patients that weren't getting better with any of the other therapies available.
So do you want to say a little bit about how immunotherapy works on a cellular or molecular level?
Yeah, so I talked about it briefly, but I can tell you a little bit more. We're basically trying to make the cells in our immune system better at detecting cancer cells. So one, in my opinion, very cool way of doing that is we can actually take cells from a patient, immune cells from a patient, from their blood. We can genetically engineer them to make them a lot better at recognizing a cancer cell. Then we can multiply them on a dish in a lab, and then we can inject these supercharged cells back into the patient, which were so it's their own cells that are just a little bit better. We made them work out a little bit and they got a lot stronger. And now they're much better at fighting the disease.
Yeah, that's actually what you're describing is called CAR T therapy, and it's really taken over oncology like a storm over the last 5 years, really, and is now the first line treatment for many types of blood cancers, leukemias, and so forth. It's really groundbreaking. And again, this kind of stuff didn't exist even 6 or 7 years ago, and it's only because people have funded cancer research, through places like Biolacura, that these kinds of things have happened. So you mentioned genetics and you mentioned environmental. So how do the two kind of interact to lead to cancer? I mean, some people have a genetic predisposition but don't get cancer, and then other people and they do get it. So how does that work?
So I need to get a little bit deeper into science, and I hope that I can explain this in a non-scientific way. But there's another layer of genetic regulation that we are still learning about, which is our knowledge of it is fairly recent, which is epigenetics. And that is where the environment can modify or genes without necessarily causing a mutation. So the structure of the gene remains the same, but now we're affecting, for example, how it gets translated or how it interacts with other pieces of DNA that are nearby. And this is a very non-scientific way of saying it, but the environment can affect how that gene gets expressed or how the protein gets created without necessarily affecting the basic structure of DNA. And so some people might have those mutations, for example, in DNA, but they haven't been exposed to the specific environmental insult that could cause that to fully turn on, for example.
I think Chris has a question. Is cancer largely a 20th century disease, or has it been prevalent throughout history? It's a great question.
Yeah, I'll give my interpretation, but then maybe you can help me out, Daryl. So I think it's been prevalent throughout history. We just weren't as good as detecting it as we are now. But I certainly think that we have a lot more environmental insults in today's world that have caused the prevalence to increase. What is your take?
Yeah, I think that's the case. I also think that our life expectancy now in the 21st century is much longer than it was 50 or 100 years ago, and so people are living commonly into their 70s and 80s, and so some of the cancers that are more common as people age are more prevalent now than they were. People didn't live to be 70 or 80 years old 100 years ago, and so prostate cancer, which commonly occurs in that age group, was very, very low prevalence. But as you point out, it's all about the toxins in the environment, things from cigarette smoking to chemicals in the environment that are carcinogenic.
Obesity.
Yeah, I mean, all of that is certainly contributing to the increased prevalence. And right now, cancer is the number one killer. Many cancers are the number one killer in different age groups, and and that's probably because of things we're exposed to. So it's a major problem. So we have another question. I think Susan's going to post it about personalized medicine. How does personalized medicine work in the context of cancer? So I guess you kind of covered this a little bit. Mm-hmm. You want to say anything more about that? So how it specifically, like for example, in breast cancer, when you're, if you're hormone positive versus hormone negative, how does that tailor the treatment towards a specific type of breast cancer, or to increase prognosis?
Would you like to tell us about that, Daryl?
Sure. Yeah. So as I think Florencia mentioned earlier, I mean, so we're getting down to cancer at a very basic molecular level, and so to the point where we're actually sequencing individual cancers. And so what we used to think of as lung cancer is now many different kinds of lung cancer, and the and you can tell which type of cancer it is based on the molecular signature pattern, and different signatures respond to different treatments better than others. And so by knowing that basic molecular information, you can give somebody the right kind of chemotherapy that's more likely to work in one type of cancer versus another. And same thing for breast cancer and some of the other common cancers. Again, we're really getting into the molecular level for some of these things. I wonder if you, is cancer contagious? Can you catch cancer from somebody by, or no?
Not from somebody who has cancer, but it could be contagious in the context of viral induced cancers. So, for example, HPV is an example of that it can lead to cancer eventually. So the cancer itself is not contagious, but what originates it is.
Yeah, and Emily earlier in a session we heard this morning talked a lot about HPV and cervical cancer and how you can prevent that with a very simple vaccine that now is recommended for most young women, and it's really done a tremendous job at reducing how common cervical cancer is. So is there a cure for cancer on the horizon, you think? I mean, what do you years ago, I guess it was Ronald Reagan, who he claimed he wanted to cure cancer. That's a big thing to try to do. So where do you see the future of cancer treatments? And
I definitely personalized medicine, because we were finding that cancer is a very different animal from what we thought it was. And now the reason why we have such effective therapies now is because we're targeting it to specific people, specific patients, specific tumors. So I think personalized medicine is one of those. But I also think the more we understand how cancer works, we can also work to prevent it. So rather than having to treat it, we can prevent it from happening in the first place.
Okay, I think we have a question. What about alternative treatments like music therapy, preventative dance therapy for cancer? That's an interesting question. So these are all kind of the alternative. Yeah. You want to comment on that?
Sure. And this is I'm not very knowledgeable in this field, so I'll give, I guess, a personal opinion or how I use logic to work through this. But I think things that can boost our immune system or can enhance our overall health will certainly be useful for us to fight cancer or any other disease for that matter. So I think alternative therapies are actually targeting those things. We're trying to make the things that are working well in our bodies work even better, so that now we can fight this invasion.
I think that's important. And there's other kinds of alternative therapies too. Vitamins and various added nutritional supplements that some of them make claims that they can cure cancer or prevent cancer. And keep in mind that a lot of these things are not approved by the FDA, and so you got to be careful. The ones that work either have been approved or have been tested in randomized controlled trials. In other words, tested against the conventional treatment and shown to be effective. So you need to be careful when you're thinking about alternative therapies. But I agree with you. Everything you said about music therapy, dance therapy, all that just boosts the immune system, reduces stress, and does a great job to make yourself healthier. And how about eating? Does eating a healthy diet, living a healthy lifestyle, can that reduce your chances of getting cancer?
Absolutely, yeah. If you're giving your body better tools to fight cancer, then yes. And if you do that by eating healthy or not consuming certain types of foods, I think that absolutely can have an effect.
Yeah, so Susie's asking about, can you get cancer from what you eat? Any examples? Can you think of any examples where there are carcinogens in the one that comes to mind for me is, I guess, charbroiled meat, when you overcook certain types of things like meat, you get some of that char on top, and that has carcinogens in it. And if you eat a lot of that stuff, you might you might.
And something else that comes to mind is some artificial sweeteners that are banned in the U.S., for example. Now, I was also a food scientist. My undergraduate degree is in food science, so I have a somewhat biased view of this, but I think a lot of the things that we think are carcinogens in terms of things that we eat, it's also because of how the testing for that was designed. So yes, we need to be careful and eat certain things in moderation, but we also have to think about how did we prove that that substance was a carcinogen in the first place.
Right. So, have you had any personal experiences with cancer? Have you, you have friends? I mean, I know you have friends, people like Lulu who asked the question, but
right.
Have you had family members with cancer or other friends?
Yeah.
So how is that? You want to say a little more about that?
Yeah, sure. I've had grandparents had cancer. I had close friends that had cancer very young. And actually, that was one of the things that reaffirmed my desire to continue my career in science. One friend in particular, she never had to have the chemotherapy because she's getting personalized medicine essentially. She takes a pill a day and that's all she needs to inhibit a particular protein that's, unregulated, and she lives a normal life.
I mean, we've all been touched with cancer. So Susie asks, why do we use chemotherapy and radiation to treat cancers? Maybe you can talk about the differences between chemo and radiation and how they work, and why some cancers are better to one versus the other.
Yes, but I will also ask that you correct me if I'm wrong. So chemotherapy, we're essentially giving cells we're targeting rapidly dividing cells, which is, as we know, cancer cells. But the reason why chemotherapy is also bad for us is because there are a lot of other cells in our body that are rapidly dividing cells. In terms of radiation, we are stimulating DNA damage in these cells, in cancer cells, which, as I mentioned in one of my slides, they are actually more susceptible to this than normal cells because they have none of those checkpoints that normal cells go through and check their DNA and say, hey, should we continue going or should we stop and fix this?
Yeah, and the most common side effects, as you point out, from both chemo and radiation have to do with the fact that they're not 100% specific for the cancer cells, right? So, chemotherapy will affect cancer cells more than non-cancer cells, normal cells, but it'll also affect normal cells. For example, it'll affect the cells in your hair follicles, which is why people lose their hair. It'll affect the cells in your intestines, which is why people sometimes have intestinal bleeding or nutritional issues during chemo. And the radiation treatments, even though you try to target them specifically to where the tumor is, they may also affect surrounding tissues, and that can cause all kinds of problems depending on where your tumor is. And so, again, all of this is reasons why research is needed to try to better target the chemotherapy toxins to the right cells rather than the wrong cells, the normal cells, and to try to target radiation treatments to the right cells versus the wrong cells. A good example of radiation targeting is well known to Matt. So I don't know, some of the people in Biolacura know Matt Bergen's story. He had thyroid cancer, and the way the treatments work for that is you give iodine, which the thyroid takes up. That's radio labeled, and so it selectively targets. There's Matt.
Hi, Matt.
So Matt can tell you about what it feels like to be on the other end of that coin. But that's a great example of where research has allowed scientists to target specific cells that are malignant using, an understanding of the underlying molecular biology and the molecular basis for the tumor has led to cures. Matt's cured now. So you want to say a little bit about that, Matt?
I wasn't going to jump on, even though cancer research is near and dear to my heart. But as soon as you brought up radioactive iodine, kind of had to jump in. It was interesting at not understanding cancer, and then walking down to the radioactive part of UNC Hospital, and then everybody else has these suits on to prevent radiation. And then I'm there in sandals and a t-shirt and walking in, and they have gloves on with tongs to grab this little pill and put it in my hand. They're like, swallow this. And I'm like, what? But I was just swallowing radioactive iodine. And then the tumor cells that I had were specifically thyroid cells. And so thyroid cells uptake iodine. And so uptakes it, kills the cancer cells. And, I'm still here today. So I'm really thankful for that. That's it.
Yeah, it's a great story. Well, yeah, again, at a high level, that's kind of the way all this personalized medicine's supposed to work, where the difference is, if you have lung cancer, we don't really know now, or breast cancer or ovarian cancer or some of the childhood leukemias, for example. We don't know how to target just the cancer cells versus the normal cells. And that's why, sometimes people get sicker from the treatments than they actually were from their original cancers. So that's why we need to do more research and that's why we're all here with Bilacura, right? So the organization raises money to try to fund basic research and clinical research to understand all types of cancer and how they work, and you need that information to be able to develop new ways to treat and prevent the disease. So if we can get maybe Susan to put up the website, so you can go to our www.bilacura.org website and read more about our mission and our vision and what our organization does and how we operate and there's the mission. The mission is to use the power and passion of dance to raise funds and support for cancer research and patient care. This is Matt's brainchild. And it's his passion, and all of us who work on with him to help realize these goals. And so you can donate on that website, or you can go to the other website where we have donation, which maybe Susan, yeah, there it is, the https://bic.ly. Who thought of it? /32JQZ9R. I think I had that almost
just a bit.ly, but if you go to Give Lively and then you search Bilacura, you can find us. This will also be posted below. As you can see, we have $4,000 of matching donations from both Dr. Rick Wojcik and Janice Wojcik, his wife, and then for 3 other anonymous donors. So up to $4,000, they'll match today. As soon as I announced it, we actually had like 4 people immediately donate. So if you donate more than $50 today, then you'll be automatically entered. I have this Wheel of Names website pulled up, and each time I see a donation, I'm putting their name in, and then during our 9 p.m. announcements, we're gonna click it, and it's gonna spin and choose somebody, and that person will win a 2021 gala ticket. So we rarely ever give out gala tickets. To me, that's like the best part of our event. That's where the only part that I've actually teared up at before. So
Florencia, you went to the gala last year, right? Was it a good experience? What was that like?
It was a great experience because you get to see both sides of what you guys are doing. The effect it has on people with cancer, and scientists like me who are there to support the fund process.
Yeah, it was very moving. I think at the end, obviously people were in tears and stuff, and because we told some really tragic stories, but many of them ended up in a positive place, which is great, and I think everybody had hope. So it was really a wonderful event. So hopefully next year, if we can get this COVID thing under control, we'll be back to a live event rather than a virtual event. But that'll be great. So you talked about the raffle, Matt. You talked about the matching donors. Anything else you want to say before we end the session?
I mean, I could sit here for the next 3 hours and talk about cancer if you want to. I'm taking a cancer biology class right now, so it's been pretty interesting. But anyways, I just want to say thank you for telling us a little bit about cancer for those of us who don't know anything about it. And we got a DJ session coming up, and then we got some more panels and presentations. I know there's some dance classes too. We have kizomba and salsa and pachanga. Look, guys, it's not the live event. It's not the same. But however, one of the great things about the virtual event is because we're all in our living rooms, we can be best artists because we're not flying them in, and they're not booked for other events, so we're able to get the best to teach classes. So take advantage of it. I've never taken a class with Shawnee before. I'm a little nervous. I hear she is awesome. So and then Charlie Garcia too, and then we have our own local instructor Kimberly and Horatio. So
yeah, it's a great event. And then we have the health disparities presentation. I think it's 7. And for those of you who want to know more about COVID and how it affects or has affected or will affect the dance community, I think we have that at 4 or 5. I can't remember, Matt. But yeah, that sounds great. All right. Thank you so much, Florencia. Wonderful presentation.
Thank you, guys.
Time out to do this, and we'll end now. Bye-bye.
Bye.